Good afternoon. So I'm Robert Semple. I'm a clinical endocrinologist and I also have a research lab. And I'm interested in translational medicine and in genetics. And translational medicine is one of those terms which is enormously overused these days. And so I define it quite strictly according to whether the research that we do actually changes diagnostic pathways or therapies for patients. I know that you've had a lot of talks over the last few months about the methodologies of genetic research. So really what I want to offer you today in my own subject area is some worked examples, if you like, of how we've applied, in particular, next generation sequencing to finding new diseases and also, I hope, towards finding new treatments for rare disease. So I don't want to dwell too much on endocrinology, the study of hormones, my own specialist area, but I do want to give you some introduction into what I'm interested in. So insulin resistance is not really a disease, it's a biochemical problem. It refers to a situation where insulin doesn't lower blood glucose properly. And it's worthy of attention because insulin resistance in the population is very strongly correlated with some of the major diseases which cause death in Western societies, including subfertility and polycystic ovary syndrome. That's a polycystic ovary you can see on the top right of the picture there. Diabetes and its microvascular complications, which you can see down at the bottom left, as well as fatty liver disease and accelerated atherosclerosis and early death from heart attacks and strokes, as well as cancer, which is increasingly being linked to insulin resistance. So insulin resistance is a common and important disease. Now, people get insulin resistance in the general population probably for a mixture of genetic and environmental reasons. So as we become obese, a proportion of us, let's say 20 or 30%, develop very severe insulin resistance, and those are the patients that we see in our clinics. And to try to understand what the inherited variants are which determine whether or not you become sick when you become overweight, predominantly genome-wide association studies have been used, and I have to say they've accounted for only a very small proportion of the heritability which we see. So what I'm talking about instead today are those red and yellow areas on this graph. So shown there in the dotted lines are the 5th and 95th centiles from a healthy population from the area around Cambridge, and that demonstrates to you that there's quite a wide spread of fasting insulin levels in the population. We're looking at patients who are many standard deviations outside the norm, and therefore we can be reasonably confident that they have monogenic disease. So I'll be talking about patients in the extremes at the top of the graph, and also some patients who are at the extremes at the bottom of the graph with insulin supersensitivity. So I think as well as simply applying genetic techniques to patients with rare and unusual syndromes, it's nice to have an overarching physiological question or a rationale for the research. So although we engage in finding rare diagnoses, what we're actually trying to do eventually is to answer some more fundamental questions about common disease. So we want to know exactly what insulin resistance is. Defining it simply by how good insulin is at lowering blood glucose is very vague because insulin does a lot of things in the body. We want to know most crucially why some people become insulin resistant and some people don't. And then even more critically, we want to know how insulin resistance produces disease. So downstream from our genetic investigations, what we aim to do is set those patients where we've found a single gene defect causing insulin resistance, and we want to study them in deep phenotypic detail and compare them to patients with common disease, and then compare and contrast. And we're every bit as interested in the differences as in the similarities. And thereby, by comparing the knowns with the unknowns, we hope to make inferences about common disease. Now, we set out to do this, and as you can infer from the bottom of the slide here, we sometimes get taken in some rather strange directions. So you'll see a variety of phenotypes as we go through. Now, when I give talks, I often borrow this from Sidney Brenner to justify my existence and my work as both a clinical doctor and a researcher. So more than 10 years ago now, and really rather presciently in his Nobel Prize lecture, Sidney Brenner more or less said, well, look, this concentration on model organism studies as the primary discovery tool in research, at least as it relates to human disease, maybe that's had its day. And actually, and this was just before the era of next generation sequencing, but he's more or less predicting it here, as you can see. As soon as we're able to harness natural human variation, then we can borrow from the 7 billion people in the world and make detailed phenotypic observations from those. I'm one of those highly trained specialist phenotypers, expensively trained phenotypers, as you see there. And his notion was that as we became better able to exploit natural genetic variation and study it, then that would be the way that we'd make our primary discoveries, and we'd go subsequently into model organisms. And this is more or less what my lab does, and which I hope to illustrate to you over the next hour or so. But it can be tricky. So one of the problems is that we are not studying binary trays. As I've showed you, insulin sensitivity varies a lot depending on how old somebody is, how obese they are, and whether or not they have other sicknesses. And so in picking the patients that we want to study, and also within a family in deciding who is affected and who is unaffected, we're not dealing with a black and a white, we're dealing with a quantitative tray and numbers, and so a degree of clinical judgment has to go on in there. There's also relatively little familial disease because insulin resistance has quite bad effects on fertility, and so it's relatively uncommon for many members of a family to be affected. So that has impeded progress in the era when family-based studies were all very important. And although we unify our research around insulin resistance, actually it is just a biochemical phenomenon, and many different underlying syndromes are associated with it, as you'll see. So you have to adopt very bespoke strategies for any given family that you look at. We know a lot about the way insulin works in cells, and we also have a large number of candidate genes, but at least we have good cellular assays for some aspects of the syndromes. I think this is my last introductory slide, and this is to give you a very simplified version of how insulin works on cells. So insulin binds at the cell surface to a tyrosine kinase receptor. You can see that at the top left. That induces autophosphorylation of the receptor, and downstream substrates stick to the receptor and are also phosphorylated. They bind in turn the phosphatidylinositol 3 kinase, or PI3 kinase complex, which phosphorylates the membrane lipid PIP2 to make PIP3, or phosphatidylinositol 3,4,5 trisphosphate. Now that only occurs in the membrane in response to this sort of ligand action, and as soon as it appears in the membrane it's able to bind and recruit downstream substrates, critically including AKT2, which then actually is the serine kinase, serine threonine kinase, which exerts the metabolic effects in a cell which are important for physiology. And to jump ahead, I can tell you that we know of genetic defects in each of the red components of this schematic, and I'll talk about several of those today. I promised you a last introductory slide. In fact, this is the last introductory slide. We've done quite a bit of work already in the patients who are well known with single gene defects, in particular those patients with insulin receptor mutations. And one of the observations we've made is that if you have a genetic defect in the insulin receptor, you are more severely insulin resistant than almost anybody else, no matter how obese in the general population. And yet, biochemically, it is a very distinct syndrome with no fatty liver, no dyslipidemia, and levels of the fat cell-derived protein adiponectin, which are utterly different compared to other forms of severe insulin resistance. So now in our diagnostic lab, we use this biochemical testing to spot which of our patients need to have their insulin receptor gene screened. And until recently, we regarded that as being absolutely clearly indicative of insulin receptor dysfunction and nothing else. So that was until we met this girl. So this is the first proband that we studied, quite characteristic of a lot of what we do, because this was a girl with a form of severe insulin resistance but no affected family members, quite common in our line of research. And she had unrelated Nigerian parents. She came along to medical attention with features of diabetes when she was about 12 years old, and she was quite quickly identified to have very severe insulin resistance. Now, curiously, this shows the appearance of her axilla or armpit and the back of her neck, showing the very thickened skin, which is a feature of very high insulin levels in the blood. So that's one of the useful visual clues we have that we're dealing with insulin resistance. You can see her family tree is very straightforward there, and only affected child of unaffected parents. Now, what was of particular interest to us as endocrinologists was that she was extraordinarily insulin resistant, with a fasting insulin, as you can see, more than an order of magnitude higher than we'd expect it to be. And yet, despite this very severe insulin resistance, And exactly like our insulin receptor patient, she didn't have any fatty liver and she didn't have an abnormal fat profile, an abnormal lipid profile in her blood. So the assumption was immediately that she would have an insulin receptor mutation. That was a rule which had universally borne out in our previous studies. And yet we studied her insulin receptor by Sanger sequencing, by multiple ligation probe amplification to look for exon deletions, and it was normal. And so we thought, well, this is exciting. This is a novel syndrome that looks like insulin receptoropathy and yet is distinct. So with colleagues at the Sanger Institute, she had exome sequencing. And this was in our early, relatively naive days when we had a tendency to sequence probands alone rather than probands in both parents, which would be our normal approach. And at the time, we got around 42,000 variants from the analysis. In other words, after quality control, there were 42,000 deviations from the human reference sequence. And that tells you that this was some time ago, because these days through the same pipeline we tend to get around 100,000 calls of variants. And what we did subsequently was fairly straightforward. We simply identified the single nucleotide variants that changed gene function or that were predicted to change gene function. At that stage we used the DB SNP database, which was still a database of common genetic variants to remove anything that was common and therefore unlikely to cause this rare disease. And we exploited some of our in-house data sets, again, to remove things which were common in that pipeline. And this is fairly indicative of what we could achieve at that stage with this sort of filtering approach. We still were left with several hundred different variants in that patient. And that, therefore, did not give us an unequivocal genetic answer. However, the human brain took over at this stage, and simply looking down an Excel spreadsheet, I was able to identify at least one gene of major interest to us. And this gene was the regulatory one of the regulatory subunits of PI3 kinase, that red complex just downstream from the insulin receptor. And the reason I was interested in this is because the genetic change we found was major. It was a truncating mutation, a nonsense mutation, predicted to cut off some of the key functional domains at the end of the regulatory subunit. What made it even more exciting was Sanger sequencing of the parents as well as the proband, which demonstrated that this was a de novo mutation. In other words, it had appeared in the proband, but not in either parent. And there was no unsuspected paternity here, I should say, as well. And because we know that de novo mutations in the exome are extremely rare, this alone raised this known insulin signaling gene much higher up in our probability. So we did the sorts of things that we're able to do with patients. We took a skin biopsy, a quite trivial procedure. It takes about 10 minutes and a bit of local anesthetic. And we grew the skin fibroblasts in culture. And we did some Western blotting to look for evidence of the mutant protein. And as you can see, there were abundant amounts of the short version of the protein in the proband, but not in controls. So not only had we found a short version of the protein, but it was plentifully expressed. We did some signaling studies because what we fully expected to find was that insulin signaling would be impaired. And what we predicted based on the mutation was that there would be a reduced ability of the mutant protein to associate with phosphotyrosines after stimulation with insulin. So the experiment on the left here simply involves stimulation of cells with insulin and immunoprecipitation to pull out complexes containing phosphotyrosine, and then a blot to look for our protein of interest in those complexes. So what I hope you can see in the three controls, C1, C2 and C3, is that after insulin stimulation we do see recruitment of the wild type protein to these complexes. But in the patient, P4.1, there's no evidence of recruitment whatsoever. So that was good. And we replicated that in an in vitro system where we overexpressed genes of interest. So this conformed to our prejudice. We thought we had clear evidence of loss of function. But then things got a bit more complicated because we looked downstream in the pathway at some of the things which insulin does in cells, in particular stimulating the generation of phosphatidylinositol 3,4,5 trisphosphate, PIP3. We did that using a mass spec assay, and what we found was that while our patient in white at the top left did appear to have a lower level of stimulation compared to some controls, in fact we got the characteristically wide range of response in the general population, and this is normal when working with primary cells from outbred people. So actually it clearly overlapped or fell in the middle of that normal range. And also, when we looked in great detail at the ability of insulin to stimulate AKT phosphorylation, we simply couldn't see any difference between our control and patient cells, even when we used a very wide dose response. So this left us a little bit stuck, because at this stage I had very little doubt that we had the right answer. We had a rare de novo mutation in an insulin signaling gene in a patient with severe insulin resistance, and yet that is not sufficient to prove this answer genetically, nor is it critically sufficient to persuade editors or reviewers in major journals that it's worthwhile publishing your work. And so we looked very hard for a second patient and failed to find one, and therefore we had to go and make a mouse at this stage using conventional gene targeting to knock in the mutation of interest. And this, as you can imagine, took at least 18 months or 2 years. You can see our founder mouse here in black. You can see some Western blots of the mouse tissues to demonstrate that we had made a small version of the protein, just as I showed you in the patient cells. And gratifyingly, both the female and the male mice were small, shown in red compared to blue. So we'd modeled the syndrome quite nicely in terms of growth, and also in a series of metabolic studies, only one of which I've shown today, we actually managed to demonstrate that the mice are severely insulin resistant as well. So this is data from a hyperinsulinemic euglycemic clamp, during which you infuse insulin and then determine how much glucose is required to keep the glucose normal. And so our mutant mice required very little glucose, if any glucose, to be infused, while the wild-type mice or mice fed on high-fat diets still require glucose. So actually, in a very expensive and time-consuming way, I think we had proved unequivocally that the mutation we identified was the cause of our syndrome. Now, the problem is, in an era of widely available exome sequencing, you're very vulnerable to being scooped. So just as we were bringing this work to fruition, 3 different groups around the world identified the same types of mutations, and indeed one of the same mutations, in a syndrome called short syndrome, which is defined by geneticists according to the dysmorphism as being of short stature, hyperextensibility of joints, deep-set eyes, Rieger anomaly, which is an abnormality of the iris, and teething delay. And they presented all these pictures in their reports of affected patients. Now, although our patients had not been labelled with short syndrome and actually didn't have any of these eponymous features, when we looked at her face and compared it to those faces, using the great integrative ability of the human eye to deconvolute human faces, we could see that she did have a mild and previously undiagnosed form of the syndrome. And the mutations found by these other 3 groups all fell in the same domain of the protein, towards the C-terminal SH2 domain, as you can see. And so this nicely defined a new syndrome, and meant that on the plus side, instead of having a mouse model of one patient, we had a mouse model of a more common syndrome. And as luck would have it, since then we have recruited 4 more patients, all with the same genetic defect and severe insulin resistance. And the only point I'll make about this is that we're pursuing the downstream metabolism quite hard, because this has confirmed that each of our patients has a very, very severe degree of insulin resistance, except one younger boy, which is not surprising, as insulin resistance is less common in that age group. And they all had insulin resistance with no fatty liver and no dyslipidemia. Now, one oddity about this, which I would point out to you, is that I mentioned to you before that adiponectin is an excellent marker of insulin receptor dysfunction. And any time we see severe insulin resistance with a plasma adiponectin level which is preserved, we are very confident that we're dealing with loss of insulin receptor function. Now, in the proband who I've showed you already, in fact, the adiponectin level is extremely low. And so that had piqued my interest, thinking that we were dealing with a 3rd subphenotype of insulin resistance. But as you can see, our other 4 patients actually had preserved or high adiponectin levels, so in every respect, they looked like insulin receptor patients. So we were a bit troubled by this discordance, until we went back again to the exome sequencing data and had a look at our proband and found that there was a second mutation, a missense mutation, in the gene that encodes adiponectin. Now, this is a missense mutation towards the end of this very complex gene, which is a structure like a collagen C1q. This is not a de novo mutation. This is a mutation that's found in the mother as well, who also has extremely low adiponectin. And you can see from a non-denaturing blot of the serum from the proband there, that there's very low levels of adiponectin compared to controls, and certainly compared to insulin receptor patients. We were able to also make and express that adiponectin mutant in cultured cells and show that mutant plasmid was able to result in the secretion of only a very low level of adiponectin. So quite independently, in our one patient, we had managed to find not only a pathogenic PI3 kinase mutation, but a fascinating mutation in the adiponectin gene, which allows us to answer a variety of questions about the role played by adiponectin in common metabolic disease. And so that's something we're pursuing in some detail. And so this is translationally useful because now we're able to molecularly diagnose patients with this syndrome, which we now recognize quite clearly. And it has also now presented with us with a series of different patients with different forms of insulin resistance. So for the first time, we can look at this morass in the general population of people with high levels of insulin called insulin resistance and some associated diseases, but where we don't know the cause. And we can compare them with a series of different forms of insulin resistance caused by different defects. And we can identify that some of those things are seen in the presence of insulin signaling defects and some of them aren't. And this begins to tease out for us where the mechanisms are, where the underlying defects lie. And it's conceptually very important, potentially, in new treatment strategies for the future. So I think that demonstrated that when it works, it's easy. That was a little bit of Bayesian intuition, a bit of smartness looking down a long list of genes. But it's not always the case, of course, and there's a huge skew in anything you see written or published towards things which were successful. And I think everybody who does a lot of exome sequencing also has a lot of examples of cases where they've looked and haven't been able to find the answer. So in a spirit of honesty, I'm simply going to show you this rather heroic tale of, so far, failure to find an underlying cause. So this is a family from Turkey, an outbred family from Turkey with no known blood relationships, where at least 8 members are affected by a very clear syndrome of short stature, severe insulin resistance, and facial dysmorphism. Now they too, they're our only other family who share the pattern of severe insulin resistance and high adiponectin levels. And yet, once again, they don't have any problems with their insulin receptor, have normal insulin receptor expression in fibroblasts, and look for all the world as if they're a third example of a novel insulin signaling defect. But in this case, as well as doing exome sequencing, we have the opportunity to do some linkage analysis of families, which, even if it isn't powerful enough to get us to a significant LOD score on its own, it's enough to refine the way that we look at the exome sequence. And so this is what we've done. A Wellcome Training Fellow in my lab, Eleanor Raffin, ran a linkage analysis in the family and found one quite large region on chromosome 19, which was significantly linked with a fairly high LOD score to the disease. And this was refined subsequently with some microsatellite mapping so that we were left with a 19 megabase interval, including 524 genes on chromosome 19. Now, in the old days, that's a little bit depressing because it's not practical to sequence 524 genes at once. But of course, in an era of exome sequencing, we can sequence the whole exome and then simply refine our search in a spreadsheet to that interval. And that's what we did. Now, the problem was, although we had good quality exome data, and although we've actually subsequently gone back and sequenced several different family members, we simply haven't been able to refine our analysis down to an individual gene. So we have achieved standard levels of coverage of about 90% of the exome in this interval. We have found 1 or 2 very rare variants, but no matter how we filter, we're unable to come up with anything that looks like a convincingly plausible biological candidate. This is quite frustrating after a great deal of work, and at present we are going back and redoing this analysis with genome sequencing with the hypothesis that we may be looking at a mutation in a regulatory interval. But a residual worry is that there is a large zinc finger cluster among these 524 genes, and I think it still is plausible that we simply haven't managed to capture the unique sequence which confers this phenotype. As you're probably aware, it's particularly difficult to capture exon 1, which is often very GC rich, so it could well be a sequence lying in there. So sometimes it doesn't work. This is a heroic tale which may yet come up with an answer. Having been very confident that we would find an answer from the start, in fact much more confident than in our previous case, we simply haven't been able to do that yet. Okay, so now something quite different. As I said, you get taken sometimes in strange directions when you start off with the rare things that happen to humans naturally. And although insulin resistance is my primary concern, we have also looked quite a lot at reciprocal phenotypes caused by activation of insulin pathway signaling. And one of the extraordinary syndromes we've looked at is a syndrome of very severe hypoglycemia in infancy. So these 3 patients all presented or were presented by their parents within about the first 4 or 5 months of life with the story that they tended to collapse and have epileptic seizures any time that their mothers tried to extend their feeding interval beyond about 3 or 4 hours. And in each case they underwent intensive investigations in hospital for many weeks, trying to understand why this happened before eventually concluding it was due to severe spontaneous drops in the blood glucose. What was further unusual about the syndrome was that the pattern of metabolic defects looked exactly as if too much insulin was being made, and congenital hyperinsulinism is a quite well-known entity. And yet, despite a metabolic profile consistent with too much insulin, there was never any detectable insulin. So for all the world, biochemically, the patients looked as if they had some insulin-like activity, but without the presence of insulin. Unusually, each of them also had some degree of left-sided hemihypertrophy. So you can see in this first and most severely affected boy on the left, there's quite pronounced increase in the size of the left leg compared to the right, and extra adipose tissue on the left side of the chest. And there's a lesser degree of left-sided asymmetry, extra growth in the other 2 patients, as you can see. You can also see the percutaneous feeding tube in that boy, and each of these patients required feeding through a surgical tube for at least the first 15 years of life to prevent them from having epileptic seizures at night. I should say that in each case, neither of the parents was affected. So again, this is one of our early examples of the application of exome sequencing, if anything, even earlier than the last case I showed. And you can see that we came up with 35,000 variants from the end of our exome sequencing pipeline. Remember I said that we get around 100,000 these days. And again, we hypothesized that this must either be a de novo mutation or a rare compound heterozygous mutation, or possibly an unlucky homozygous mutation, as the parents weren't affected. And so we went through the same iterative process that I showed before, and still, again, we're left with several hundred genes of potential interest. Once again, the gap was bridged at this stage by the human brain and our a priori knowledge of insulin signaling. And looking down a list from the first patient, we found out that there was a missense mutation in AKT2, that classical insulin signaling gene. Now, the missense mutation altered a very conserved glutamate in the plextrin homology domain, very exquisitely conserved among orthologs and paralogs. And we actually, on sequencing of our first proband and the other 2 patients, found that all of them had the same mutation. And in each case, it was de novo. And in fact, in our 3rd patient, it was also mosaic, as you can see from the middle chromatogram there, compared to the bottom one, which was characteristic of the other 2 patients. So according to the same arguments which I used before, the rarity of de novo mutations, the very strong candidacy of this particular mutation, we were pretty sure we had the answer. And critically in this case, because we had 2 independent patients, it was pretty well a done deal. That's simply to remind you where AKT2 falls in the signaling pathway, and to demonstrate that, as predicted, the plextrin homology domain mutation, which allows the protein to approach the plasma membrane even without the requirement for phosphatidylinositol 3 kinase, does lead to a low level of basal activity in patients' fibroblasts. And when we expressed the wild type and mutant protein in cells, we found that although in the wild type cells, as you can see in the top row of the micrographs, there is nuclear expression of the transcription factor FOXO1 in the basal state, which is excluded from the nucleus in response to insulin. The mutant AKT2 is able to exclude that key transcription factor from the nucleus even in the absence of insulin. And we predict that that will result in a failure to turn on hepatic glucose production at the time of fasting. So it very nicely agreed with the phenotype. And this has actually been part of a wider story, which I just commend to you for general interest. So there are 3 isoforms in the AKT family, a very fundamentally important class of serine 3-enane kinase. I've described to you the metabolic phenotype causing hypoglycemia, with some degree of asymmetry, which we still don't understand. The AKT1 isoform is much more expressed in somatic tissues and is believed to be more related to the growth. And Les Bieseker's group at the NIH has beautifully demonstrated that the horrible, progressively deforming condition Proteus syndrome, which is also suspected to have been the condition affecting the so-called elephant man, Joseph Merrick, is caused by the very same mutation, the homologous mutation in AKT1, as you can see here with deforming skin and bone overgrowth. The AKT3 isoform is predominantly expressed in the brain, and in keeping with that, the very same mutation in AKT3 leads to asymmetric progressive brain overgrowth, which you can see in both these scans on the right-hand side, each of which led to the need to remove one of the hemispheres of the brain, which is what you see on the right-hand side of the image due to intractable epilepsy. So it's a very nice allelic series of the same mutation in different homologous members of the family. Actually, in this case, beautifully in accord with prior mouse knockout studies. And this very much got our eye in, or attuned us to the notion that there might be other activating mutations in the same pathway. And fortuitously, around the same time, we met this lady. Now, she was sent to us because one of her doctors has suspected she may have an abnormality in fat development. And we looked at her and took a different view. Now, looking at this series of images from her early childhood, you can see that from early life she had a gaunt, somewhat emaciated upper body, but progressively enormously growing legs, which has continued all her life to her current age. And you can see that ultimately she has a muscular, thin appearance in her upper half, enormously enlarged legs, predominantly accounted for by fat. And she's also had to have an amputation of her left leg below the knee because of intractable infection in an ulcer. Now, if you look at the image in the middle on the bottom, you can see that there's evidence of clear asymmetry. So although she has lots of fat on the right side of her abdomen, there's a clear demarcation and almost no fat on the left side. So looking at this in clinic, I find it very difficult to conceptualize this in any way other than the presence of a mosaic mutation. And so we did a very simple thing. We did a punch skin biopsy from this lady's arm. We grew some dermal fibroblasts from that, and we did the same thing from her leg. And then we ran exome sequencing of both leg and arm and asked the question, are there mutations present in the leg which aren't present in the arm? And we actually got lucky for reasons which I'll explain in a second. So we found the usual large number of 50 to 60,000 variants in both the arm and the leg samples. We refined those to only very rare variants and found that around 1,400 of the rare variants were found in both. There was a more or less symmetrical discordance, with some variants being called as present in leg but not arm, and vice versa. We tidied that up by, first of all, Sanger sequencing, which made some of the variants drop out, and then by reculturing some very fresh fibroblasts, because we were concerned that the samples that we'd originally used had been from fibroblasts in culture for some time, and they may have acquired somatic mutations there. And so we ultimately concluded we could only find 2 mutations which were specific to the leg, and one of these was a classical activating mutation in PI3 kinase, which we were very convinced was the answer. And you can see that when we looked at this directly with Sanger sequencing in affected leg cells, the mutation was clearly present, but absent in the arm. When we went back to tissue specimens from the amputation, you can see that, however, the mutation was present at much lower burdens, consistent with somatic mosaicism. And in fact, I think this is where our luck came in on this occasion. Because we had used cultured cells, we had ex vivo selected for the mutants, and we simply used Mendelian calling algorithms, and that's why we'd found that apparently heterozygous mutation in the leg. If we had worked from tissue samples, that may not have been called at that low level of mosaicism, and we probably would have had to adapt our algorithms to pick up lower levels of abnormal reads. Now, the reason we were so confident this was the answer was because this is a very well-known mutation from the cancer world. This is a histogram taken from the Sanger Institute-hosted catalogue of somatic mutations in cancer. And across the coding sequence of the PIK3CA gene, you can see there are 2 very striking hotspots where mutations are found in cancer, one of which is right at the C-terminal of the kinase domain, which is where we had found this mutation. We used a similar approach to what we had done before in the AKT cells, and we demonstrated that there was clear basal activation of downstream signaling even in the absence of ligand. And we also found that PIP3 levels, the substrate, so the product of PI3 kinase, were elevated in the basal state to about the same level as seen with stimulation of normal cells but with ligand. So by several different assays we showed biochemical evidence that there was increased PI3 kinase signaling in these cells. We duly found a handful of other fairly similar patients and published that now a couple of years ago. But around the same time, many other groups around the world were thinking in similar ways and adopting similar approaches for other syndromes featuring overgrowth of different parts of the body. And the world has moved very fast since then, and to cut a very long story short, it's now clear that there's a very wide range of syndromes of so-called segmental overgrowth, and that refers to asymmetric progressive overgrowth, which previously had been classified by phenomenologists, by clinical geneticists, according to which part of the body, for example, was affected. But it's become clear that these probably all share the same genetic basis in the form of activation of the PI3 kinase AKT signaling pathway. And so you can see now a snapshot of that disease spectrum where, unlike in Mendelian disease, where the phenotype is generally a product of disease genotype, which is commonly the dominant feature, and genetic background and environment, in the case of these disorders, while the disease genotype may play some role, really the key thing is the precise time and place of the founder mutation. And these are probably mutations happening very early in embryonic life, around the time of gastrulation. And you can see that the commonest patients simply have enlargement of a single finger or a hand, progressing through to the very severely affected lady, which I've shown you already. And sometimes, even when the overgrowth quantitatively isn't very severe, it's catastrophic functionally because it affects, for example, the face, as you can see in this poor boy here. And so the work has all of a sudden opened up this series of conditions which have had very few treatments apart from intermittent surgery up until now. And a huge number of scientific questions and also translational questions now arise. So we want to gradually, for scientific reasons, try to work out the timing of the mutational event and which cell of origin and which lineages are affected. We want to know if there is a genotype-phenotype correlation, and we want to understand whether all the growth is cell-autonomous or whether there are paracrine effects as well. But the critical translational questions for these patients are whether or not we can predict what's going to happen to the overgrowth. Is it going to progress inexorably or will it stop? Can we distinguish which bits of overgrowth we can reverse and which bits are actually early life consequences which are by now irreversible? And also, given that these are classical cancer genes, is there a cancer risk? And I can tell you that it looks as if while there may be a very small excess risk of cancer, it appears to be very small in these conditions indeed. But perhaps most exciting of all, and this is very rare in genetics, generally finding the genetic basis of a novel condition is exciting because it allows family screening, it allows explanation to patients of what the problem is, but very rarely does it actually lead to application of an off-the-shelf treatment. And we're nearly in that situation here. Because the world of cancer and the drug companies interested in cancer have been working very hard to inhibit this pathway for the last 7 or so years since the same mutations were found in cancer. And so at multiple levels of the pathway, inhibitors exist, including mTORC1 inhibitors, which are already established in clinical use. So a very obvious question for us was, would we simply be able to take some of these tablets, whether in clinical practice or experimental, and effectively treat our patients? And there is some intimation from related patients in the literature that this might be the case. So far we've done fairly straightforward things. We've taken cells from our patients with active signaling, and we've put some sirolimus or rapamycin on the cells and shown that we're able to turn off the basal activation with exquisite ease using very small doses of sirolimus indeed. So you can see the phosphosignal being turned off on the left-hand side of this image. You can see that quantified with ELISA on the top right, and you can see how sensitive it is in terms of dose response on the bottom right. Not only that, but on the grounds of clinical need, we did start some after careful discussion with our patient and her primary doctors. And she started a very small dose of sirolimus now some 4 years ago. And this shows the data over the first 3.5 years of study and demonstrates that she has lost an enormous amount of weight to the extent that she has gone down to one of her much earlier prostheses. She's had to have a lot of her specialized shoes remade. She's improved her mobility, and she's very, very pleased indeed with the outcome. Now, this is just a single anecdote, but we're now on the point of designing a pilot study of sirolimus in similarly affected patients. And our hope is, at least for the fatty component of these syndromes, we may be able to reduce fatty growth to a very significant degree and get significant clinical benefits that way. And that's something we're planning to roll out to a wider spectrum of the patients, and also to investigate the specific PI3 kinase inhibitors in collaboration with pharma. So that concludes the work that we've done on insulin signaling itself. But one of the joys of working in human genetics is you sometimes get surprises and go in very strange directions. So insulin resistance isn't always caused by problems with the known insulin signaling pathway in cells. And particularly through study of recessive diseases, you can get unexpected mechanistic insights. Now, this started off, actually, this was the very first patient we applied exome sequencing to. And this was the only affected member of a family whose parents were consanguineous, as you can see here, where there were a number of other disorders within the family, but nobody else with the syndrome that she had of growth impairment with a small head, bilateral drooping of the eyelids, and severe insulin resistance. Now, because we were confident that she would have a homozygous disease, we had much more power to refine our candidate gene list just to look at rare homozygous variants. Again, in an individual patient, it's very rare indeed to get down to anything more than a handful, less than a handful, and we cut things down to 24 mutations at this stage. And actually, in some ways, it was disappointing because we found an obviously pathogenic mutation in a known disease gene, which was the Werner's DNA helicase. However, actually, this was of significant value to this girl because Werner's syndrome is a form of premature aging which normally can't be diagnosed until the 20s, when premature atherosclerosis and graying of the hair and cataracts and the like appear. And in fact, we had diagnosed this when she was in her early teens, probably a decade before the clinical diagnosis would have been made. And even going back to her at 16 years, we saw some of the characteristic tissue changes of Werner's syndrome. You can see that having been quite chubby at the age of 7, she's progressively thinning and losing both muscle and fat from her arms, and that generally progresses to severe joint contractures. And you can see some thinning of her hair on her head as well. And so because of this finding, we've been able to give her high-dose vitamin C, which has been suggested in animal models to retard some of the aging process through reduced reactive oxygen species. And she's also in an appropriate surveillance program. But of course, what we ultimately aim to do when studying rare unknown things is to find new biology. And that's what we found in the very last case study, which I'm going to show you. So at present, the lady that we studied is 25 years old. But right from when she was born, it was clear that all was not well. She was unusually small, with a proportionately small head, and a slightly unusual face with a prominent mid-face and a small lower jaw. But she was otherwise well. Her parents were clinically well, as were her 4 siblings. Her parents, both Europids from North Wales, were also not related to each other. Now, this is a slightly unfair image in that I am 1.95 meters tall, so many people look somewhat short next to me, but you can see the spectacular degree of growth impairment of this lady in her adult life. This is her when she's 24 years old, standing next to me. And so her syndrome was one of what's called primordial dwarfism, in other words, severe growth retardation that is apparent from the earliest stages of development in the womb. She also developed very severely insulin-resistant diabetes and fatty liver from the age of around 9 or 10 years old, which is why our interest had arisen. And yet there was no discernible problem with growth hormone or other hormone signaling pathways. The only other clue was that there was some evidence of impaired DNA damage responses. She had no evidence of Bloom syndrome, the prototypic form of primordial dwarfism with abnormal DNA damage responses. But persistently, at several stages of her life, there was evidence of an excess of damaged chromosomes and chromatids. However, that seemed to be reducing with time. It was not diagnostic of any particular disorder, and the tentative conclusion at the time was that there had been some clastogenic insult, a DNA damaging insult in early life, which had gradually disappeared as the lymphocytes had grown older. So after a due search for the genes known to cause similar syndromes and a few speculative attempts to be clever and to sequence potential candidate genes, and a search for pathogenic chromosomal microdeletions, we also went for exome sequencing of the patient, and being wiser by this stage, also both parents at the same time. And because the family was not inbred, and because there were no other affected members, we reasoned that this must most likely be a compound heterozygous problem, although we knew this could also be a de novo mutation, or possibly an unlucky homozygous mutation. And we analysed things in that spirit. And this is really a very powerful approach in this setting with a severely affected child in isolation. So as you can see, we managed to find somewhat more variants than we had done when the pipeline was a little bit younger. We were able to exclude a large number of things based on the much bigger data sets of control populations by this stage, and to refine things down to only 3 different genes. So these were the only 3 genes we were able to find which had 2 highly likely loss of function mutations, because we weren't able to identify any convincing de novo mutations from the exome sequencing. So we had 3 genes for a variety of reasons. Having dug in the literature, we thought that the NSMCE2 gene was most likely, but the other 2 genes remained there stubbornly as well. One of the reasons for favouring NSMCE2 was that when we looked at the much bigger data sets, as I said, of control variants, there was really hardly any evidence of these variants in the NSMCE2 gene, although one of the variants was found in a couple of patients in the 13,000 or so available through the NIHR BRC exome variant server hosted in Seattle. While the variants we found in each of the other genes were found at an appreciable frequency across all populations studied. Now, of course, when dealing with rare recessive disease, that doesn't rule them out, but allied to prior functional studies, we believe that NSMCE2 was the answer. But again, that wouldn't satisfy reviewers at this stage. We went back to the family. We confirmed that only the proband was compound heterozygous. All other family members were either wild type or heterozygous for one of the lesions. And each of these frameshift mutations affected the same gene, as I said, the sumo ligase, NSMCE2. One of them cut off the entire catalytic domain, the SP ring domain, while the other one occurred right at the end of the protein. Now, the key thing was that we had one other patient who had some similar features. So this is a Parisian lady, also less than 1 meter tall in adult height, with severe primordial dwarfism, very severe insulin resistance, and fatty liver and pancreatitis due to her high levels of triglyceride. So we didn't exome sequence her, we sequenced NSMCE2 directly in her, and extraordinarily we found the very same 2 variants, which troubled us quite considerably, mindful of the possibility of contamination in the lab in any PCR-based analysis. However, we were quite convinced in our own procedures, and even more convincingly, we independently had parental samples sent to us from the lady from France, the second lady I showed you, and we confirmed that both her parents were heterozygous for the mutations. And in fact, subsequent haplotype analysis suggested that both these ladies from the westernmost edges of Europe probably shared a common ancestor around 4,000 years ago on each of those alleles. So we think this is an ancient Celtic founder effect. And this is where things for simple insulin signalers became somewhat complex. So NSMCE2 encodes a sumo ligase. Sumo is a small peptide, like ubiquitin, which is used to modify genes as a means of gene regulation. And it's only one component of a much bigger complex, a multi-subunit complex, which is similar to cohesin and condensin. These are much better understood complexes involved in various aspects of chromatid cohesion and condensation during cell division. We did fairly simple things first, and we blotted for NSMCE2 in patient fibroblasts and found that we could see practically none in the patients. And what you look at here is increasing amounts of protein being blotted from left to right in each schematic. So very little protein in the cells. We were able to show defective autosumolation by in those cells, which is not surprising given the lack of protein expression. But we also found a number of markers of DNA damage. So these are surrogate markers which are widely used in the DNA damage world. In the images at the top, you can see on the 2 left-hand images 2 micronuclei. So these are inappropriately packaged up little fragments of DNA containing chromatin. And we can see a nucleoplasmic bridge on the right, which is a persisting strand of nucleoplasm between two nuclei after mitosis. So all of these things are indirect evidence of an incomplete DNA damage repair during the process of mitosis. And when quantified through counting, we could show that in our patient cells, these were highly significant and exacerbated by hydroxyurea, which depletes deoxynucleotides. Now, what I enjoyed most about this study was our subsequent use of a lentivirus to re-express either wild type or sumo ligase dead NSMC2 in these primary patient cells with recounting of the scoring of the markers I showed you before afterwards. And this showed that when we added doxycycline to the cells, because these were conditional lentiviruses under the control of doxycycline, when we acutely turned back on wild type expression, we completely suppressed the evidence of either nucleoplasmic bridges or micronuclei. So you can see that in the pale grey bars on both sides. But when we turned on a version of the gene which had no sumo ligase activity, actually we made things much, much worse. So this was another way of trying to add value to a rare patient study by genetic complementation using lentiviruses and other tools at our disposal. So we were pretty convinced that we had the right answer. And through collaborators at the Sanger, we were also able to use zebrafish and show that with morpholino-based knockdown in very early stage zebrafish, efficient knockdown of the gene resulted in severely dwarfed fish. So zebrafish are wonderful models for some simple phenotypes, such as organ development and growth. Not only that, but in those fish with the morpholino knockdown, if human mRNAs were re-injected, we showed that we could completely rescue the body length size if we injected wild-type mRNA, but not if we injected either of the mutants that we had discovered before. So again, in a model organism, this was a very nice proof that both of our mutations in isolation were pathogenic. And so really, those were just a series of case vignettes to show the types of approaches and types of discoveries we've made in the arena of insulin resistance. The wider sort of 30,000 foot picture of insulin resistance looks something like this at present. So I've showed you patients with mutations in PIK3R1 encoding one of the components of PI3 kinase, and we know that other defects in the insulin signaling pathway also produce severe insulin resistance. The biggest single gene, the single mechanism underlying severe insulin resistance, however, is not to do with insulin signaling, but rather it's to do with fat cell development and function. So on the left-hand side here, each of these disorders, some of them recessive, some of them dominant, cause severe insulin resistance by impairing fat cell development, which means that we have an inability safely to store the excess of calories which we eat in adult life. The last case I showed you touched much more on this range of very fascinating complex syndromes down on the bottom right, where severe insulin resistance is only one component of a disorder which features, for example, severely impaired growth or other multi-system features. And there is beginning to emerge some common themes from this. So there are several different disorders of DNA damage repair or mitosis or centrosomal function in this group of disorders. We would only know about each of those things from human genetics, and I think it's opened up a whole interesting area of understanding how some, and only some, primary disorders of mitosis and DNA damage repair can produce severe insulin resistance. My personal prejudice at this stage is that fat cell precursors are selectively damaged or are selectively susceptible to some forms of DNA damage. And in the face of these genetic defects, that tissue particularly suffers as life goes on to produce a sort of acquired lipodystrophy. But that, I should say, is unproven. So we have been tremendously excited by exome sequencing applied to our collection of rare patients. For many, many patients, we have resolved things down to several tens or hundreds of variants, but because they have isolated syndromes, we may well be looking at the mutation that causes it, but we don't yet know that. I've skimmed from the top the ones which were more obvious to us, either because of genetic luck or because we had good reasons for suspecting the mutated genes already. I've shown how you can either find novel defects in known genes and then learn lessons from physiological study of those patients, or sometimes you can quite quickly apply experimental treatments in other cases, especially in the case of rare recessive diseases, the trajectory is not so much towards early treatment, but rather towards completely novel directions and novel insights in pathogenesis of common disease. But I think from all the work I've showed you today, if I had to take one bit, my biggest excitement at present is around the activating mutations in the PI3 kinase pathway, because I suspect that within 3 or 4 years of finding those mutations, we will have new tablet-based treatments for patients who up until now have been treated only intermittently by the surgeon's knife.